Okay, everyone. We're slowly coming back. Hopefully that break was welcomed by, it was welcome for me. So thanks for allowing, for giving us that time. Okay, I think we'll get started with our second session this morning. And in keeping with the theme of this conference, which is practical solutions and success stories, we have a couple of talks coming up on what we would classify as some practical solutions. So our first speaker this morning is, or this session, is Dr. Amy LeBlanc, and she is a board certified veterinary medical oncologist, clinical trialist, and translational molecular imaging researcher. She currently serves as senior scientist and director of the Comparative Oncology program, which is an intramural program with the National Cancer Institute, with the National Institutes of Health. Her research investigations have focused on improving drug and imaging agent development outcomes for humans with cancer, primarily through clinical studies in pet dogs and cats who have spontaneous cancer. Her clinical oncology experience, coupled with an understanding of cancer biology and translational oncology, has proved important for several collaborative publications. Amy is going to speak to us this morning about how dogs are helping us understand cancer. So thank you for joining us today, Amy, and the session is yours.
Great, thank you. I assume the audio is okay? Everything sounds all right?
Yes.
Okay.
We hear you.
Excellent. We're still getting used to all of this technology, right? Okay. So this is the topic of the day, is how our companion animal dogs are going to help us understand cancer within them, but also hopefully within ourselves as well. So thank you very much for the introduction and the opportunity to speak today. Pat, Catherine, Camilla, everybody, very much appreciate the invitation and the assistance in getting this going today. So excited to share a few stories with you about what we're doing here within my program at the NCI. So, this is just sort of an interesting slide that makes us think, we are a lot like our pets, probably in more ways than one. And more, I think, as certainly within the family structure, dogs have really elevated themselves to a part of the family, much more so than they were probably a few decades ago. And we realize more and more as they share our environment and our home life, how similar that we are to them and they are to us in many ways, more than just companionship, but also with an eye towards the conditions that they develop naturally through the course of their lifetime, very similar to what happens to us. And to that point, there are remarkable similarities in the cancers that develop in dogs and humans. This is a nice paper that was published by Josh Schiffman and Matthew Breen several years ago now, but incredibly relevant still today, that there are over 4 million diagnoses of cancer in dogs every year, which eclipses the human diagnostic rate by far. And the types of cancers that are developing in our pet dogs share a lot of histologic and phenotypic and molecular similarities to those that develop in people. So these are just some examples listed. Brain tumors, specifically gliomas, that appear based on very recent literature to mimic childhood glioma rather than adult human glioma, which is interesting. Melanoma and many of the blood-borne cancers, leukemias, lymphomas, malignant melanoma, and then solid tumors of the breast, bladder, prostate, and bone. So we definitely have a growing appreciation for this, not just as a medical problem for the companion pet dog population, but also as a translational opportunity to advance knowledge for humans. So just to hit and reiterate a few points on that topic, that within the US, there are probably about 78 million dogs that live in our households. And this is an old statistic. This number of 1 million will develop cancer each year. That, as I've mentioned on the previous slide, that's probably more like 4 million. It's a little bit tough for us to know because we don't have tumor registries that are updated and kept with rigor that they are on the human side. But suffice to say that there are a lot of dogs suffering from cancer in this country and abroad. And many of those cancers that develop are similar to that which humans develop. And really what's come to the forefront over the last 10, 15 years is that pet owners are very, very motivated to investigate and apply very advanced therapeutic options for their pets. So what they expect for themselves is oftentimes what they're expecting for their pets. So advanced surgical and radiotherapy techniques, access to chemotherapy, access to small molecule inhibitors of altered signaling pathways, immunotherapy. There are lots of examples of where the drug development community has really risen to the occasion to provide some of these very advanced and technically complicated types of treatments that are tailored actually for pet dogs and to some extent also pet cats. So when we're thinking about including the pet dog with naturally occurring spontaneous cancer into studies of cancer biology and drug development, we really have to acknowledge some of the real obvious advantages there, that there's greater synteny between dogs and humans than there is between rodents and humans. They have a lot of shared gene expression alterations, and to some extent also many of the same molecular pathways are altered between many common canine and human cancers. And to that point, they have a very much a common druggable target landscape and similar pathways of angiogenesis and apoptosis, resistance to therapy, the natural development of metastasis and tumor progression are all observed with high regularity and frequency in dogs. And when we think about designing studies to evaluate new therapies in dogs with an eye towards translating that data for human benefit, one of the things that becomes really important is that you have access to a patient population for which you don't necessarily need fully GMP material. So often within a drug development strategy, we can enter into some of the interim steps before a fully GMP product is available for human trials, and really help optimize those follow-on human trials, not just with respect to dose and schedule, but also for responding biomarkers and other sort of deep biologic endpoints. And then also you recognize that in dogs, they often are, they recapitulate a spectrum of patients all the way from treatment naive cancers all the way to heavily pretreated cancers. So you have definitely within the context of clinical trials to offer new and investigational agents to pet owners for the very first line of treatment for their pet. You don't have to necessarily exhaust a standard of care or multiple accepted regimens of therapy before you offer an investigational agent. And we'll get into some of that within the examples I have in this talk today. And then obviously, you know, pet dogs, there's a huge variety in size and shape and all of that. And the breed idiosyncrasies and nuances between breeds are one of the really interesting areas of research right now to sort of understand what impact that might have on the data that we generate. But their body size is very relevant and is supportive of, you know, imaging and treatment, the application of treatment modalities that are often used for people. And then you have that opportunity for repeated biologic sample collection within the same patient. And all of that occurs within the context of routine veterinary care. So it really is very much a win-win and a nice complement to the other models of human cancer that we're all very familiar with. And so this is somewhat of a famous figure from Nature Reviews Cancer several years ago. But again, it sort of drives home where we think and we can show that the tumor-bearing pet dog can contribute very early on and even in later steps of human drug development in a variety of ways. And so the obvious strategies that are an advantage here are that you're working with a naturally occurring tumor that's co-evolved with its host. It is an immune competent host that has been exposed to all of the same environmental exposures that you and I have been exposed to. They sleep in our beds, they eat food off our tables, they live with us. They're not in a contrived, you know, controlled environment per se. And they really don't have, in many cases, a sort of standard of care. So pet owners always have that option to say, look, you know, I don't know that I really want to go down the traditional route, but I don't want to necessarily immediately euthanize my pet after a diagnosis of cancer. Is there another option for me to contribute to the body of medical research that we're all, interested in contributing to? And maybe there could be some benefit gleaned for us as a pet owning family. And so several years ago now, we actually were fortunate to have a National Academy of Medicine workshop on this topic. And one of the things that this meeting did was really, to a point that has been made earlier in this meeting yesterday and today, it really helped open up an agenda for what comparative oncology needs to do to maintain and build on its relevance and importance in the cancer and really in the scientific community as a whole. And one of the things that has come out of this meeting is a real emphasis on the gaps in our knowledge. The biggest gap out of this paper and of this meeting was widely acknowledged to be the lack of really deep molecular understanding of canine cancers and how those molecular changes relate to their human counterparts. And so we are actively in my program undertaking one facet of this in osteosarcoma. And so I'll get to that kind of later in this talk. But I think that if you look at the comparative oncology literature, especially over maybe like the last 5 to 8 years, it has really improved. And I credit this meeting and the work of so many people from our community towards that goal to really get a deeper appreciation of what dogs are developing and how relevant and meaningful and informative it can be for human cancers. And so when you think about what's going on in comparative oncology in North America, it is an incredibly diverse and active community of scientists and clinicians, pet owners, and just an amazing amount of philanthropy and funding has really come into our community in the last 3 to 5 years. So not just what I do at NCI, but also many, many talented people within other NIH institutes and many academic institutions represented here in this meeting and beyond, studying the opportunities and really coming up with very compelling stories about how if we look very closely at and involve pet owners and pet dogs with cancer into these studies, we can really advance our knowledge to benefit everybody. This is just a list of clinical trials that our program here at NCI has been involved in. I show it to you only to sort of touch on the point that we have the ability and the flexibility to sort of work across multiple different platforms of treatment types. So, if you look at this list, there are small molecules, there are investigational agents that are first in class. We've got a couple of examples of good immunotherapy trials that have been done over the last few years. We've done medical device work as well. And so, I think that that's one of the things that, if anything, out of this meeting, you come away with the idea that there could be lots of opportunities to impact research, not just in cancer drug development, but also in other methods of medical research as a whole. And really, to that point, one of the things that is a huge advantage of working with dogs is that you have so many opportunities to interrogate them from a sample collection standpoint. And again, all of this occurs within, routine veterinary care. So blood sampling and tumor biopsies, repeated tumor biopsies in some cases, that you can assess the longitudinal changes of a tumor over time. Looking at how the tumor tissue of interest matches or doesn't match some of the normal tissue that's adjacent to it or at a distant site. And then serially collecting samples of serum and blood and plasma and bone marrow and things like that can very much be with an eye towards, ethical management of your patient and control of pain and minimizing these sampling time points to ones that are really the most scientifically critical. All of that is very much possible within the framework of what we do. So I'm going to tell you 2 quick stories today about what we've done just in the last few years, a couple of examples of where dogs with naturally occurring tumors have really meaningfully impacted the drug development path of a couple of different agents. This first story has to do with canine melanoma and an interleukin-12 based immunocytokine. So just to recap briefly, this is a big topic, but melanoma in humans and dogs has a lot of very striking similarities in its molecular landscape and its biologic behavior, but it is fundamentally somewhat of a different disease. In humans, this is very much a dermal skin disease related to sun exposure. In dogs, this is generally a disease that occurs within the mouth, the oral cavity, so the gingiva of the lips, the gum, the hard palate, the tongue, or of the nail bed. And so despite there being probably very different oncogenic drivers of this disease, they have a pretty striking biologic convergence. So they both, humans and dogs, experience very aggressive biologic behavior and metastatic progression. And both forms, human and canine melanoma, are really poorly responsive to cytotoxic chemotherapies, but are very responsive to immunotherapy. And they have a lot of similarities in some of the downstream pathway activations. They have a general loss of some of the tumor suppressor genes that we often think about for cell cycle control. So there are some really interesting similarities, and that theme of convergence, where the initiating events might be quite different, but what you see clinically really kind of comes together very similarly in both species, is a sort of a theme of comparative oncology. And so to that point, we actually worked with this agent. This is a publication from a few years ago now. If you're interested in the whole story, I don't have all the time to tell you every single facet of this work, but we did a clinical trial with this agent called NHS-IL-12. It's an immunocytokine, a fully human IgG1 targeting antibody that was linked to IL-12, and with the intent of delivering IL-12 to areas within the tumor that had active necrosis. It had a binding motif that was recognizing necrotic tissue. So we enrolled dogs with melanoma and other solid tumors into this trial. And so the picture on your screen shows a really obvious response, a measurable response in one patient that received repeated cycles of this drug. One of the things that's really important about this trial is that you see from this study schedule here that we actually collected just a lot of really relevant biologic data from each patient over a relatively short period of time during each cycle of exposure. So there was a pre-treatment evaluation, and then on day 1 of drug treatment, in this case, this data pertains to this agent being given subcutaneously, but we also did some intratumoral dosing and explored that aspect as well. But we collected, peripheral blood mononuclear cells and did multiplex cytokine panels and looked at PK of this agent. So we measured IL-12 and other downstream cytokines to look at the pharmacodynamic effect. And we did that repeatedly throughout this trial, and we were able to also collect pre-treatment and post-treatment biopsies from the tumor itself to look at not just the tumor's immune environment, but also draining lymph nodes to see how immune cells were trafficking in and out of regional lymph nodes and how that would change during treatment. So dogs that received this agent subcutaneously achieved measurable drug exposures. And this was a dose escalation study, so we were starting at 0.4 mgs per meter squared, and each of these 4-digit numbers is an individual patient. So you see how we went through different cohorts of administration, and some of the cohorts were expanded in response to adverse events that were noted by our investigators. And then we went through an attribution and a grading process to really understand if the adverse event was related to the drug or a study part of the study or some other, or disease or some other reason. So that's why there are different numbers of dogs in each dosing cohort. But what these graphs show you is that you can indeed administer this agent and then have measurable levels within the serum, and that it is a dose-dependent effect. And then looking at the pharmacodynamic response to the agent, we were able to document interferon gamma and IL-10 release in these same patients. At several time points post-administration were assessed across several different dosing cohorts. So again, you see the dose response there for the PD effect. And yeah, so that was the interferon gamma. This is the IL-10 data. And so, when we finished this work and said, we were able to, in a dose escalation format, identify an MTD, we were able to safely give this necrosis-targeted IgG human antibody conjugated to IL-12 safely and repeatedly to tumor-bearing dogs. We established PD biomarkers that were very relevant to the human counterpart that was destined to receive this agent. And we found an efficacy signal in dogs with measurable, bulky melanoma, which is notoriously treatment resistant. And then we were able to, through the reconciliation of adverse events, relate those, as you might know, if anybody's got a background with IL-12, know that cytokine release and self-limiting lymphopenia, fever, hepatic enzyme changes would be expected. And we did identify very serious events above a certain dosing strategy. So we were able to come to this MTD of this agent and generate all this data. And so when we did that, so this is several years ago now, but when this all happened, the company said, we really think that this is now worth reprioritizing this agent. And I think that if they hadn't, this is what I've heard, if they hadn't actually received this data, I think that this agent may not have moved forward into phase 1 work. And so Dr. James Gully at the NIH opened the IND for this agent in 2011, and they moved through the standard dose levels of human patients to identify their MTD at 16.8 micrograms per kilogram. Remember, let me just go back a slide. Our MTD was 25.8, so very close as far as the MTD was defined. They had 59 patients enrolled before they could identify that MTD. The adverse event profile that they noted was very similar to what we noted, and they were able to design a lot of their PD biomarker and sampling strategies off the timing of these sample collections from what we had generated in dogs. And we generated really a comparable data set in 18 months for less than $300,000 in clinical management budget. So it was a nice story because it really led to then the reprioritization of this drug. And now recently this first in human phase 1 data was published, and this agent now is moving forward as a combination immunotherapeutic strategy in humans. So next, I'm going to run through some of the canine osteosarcoma data that we're working with now with an eye towards targeting the naturally occurring metastatic progression that occurs in that disease. So switching gears to talk about osteosarcoma for a moment, this is a very common canine tumor. It affects at least 10,000, probably many, many more dogs than that in the US every year. When we approach a patient like this in the clinic, the standard of care currently is amputation followed by some form of adjuvant chemotherapy. Typically, that's a platinum-based regimen. Some clinicians choose to combine platinum agents with doxorubicin. But however, having done all of that, we still only arrive at a long-term, either quote unquote cure or long-term tumor control. So that's 3 years or longer in my book, and only about 5 to 10% of the patients. And so they have a very short median survival time despite really aggressive treatment of only about 10 to 14 months based on the previous published literature. And that's because the majority of them are developing metastatic disease, typically to the lung, but also to other bones. When you look at how similar that is to the pediatric situation, there are some pretty important differences to consider. One is that this is a very rare disease in humans, and it's an adolescent, pediatric, young adult malignancy that probably affects somewhere around 800 to 900 kids a year. Children undergo neoadjuvant MAP-based chemotherapy, so they're going through methotrexate, doxorubicin, and a platinum, cisplatin typically, and then after chemotherapy, they go through repeated surgeries to preserve function and cosmesis and to salvage the limb that has been affected. However, having said that, only 70% of these kids really have a long-term survival. So that leaves us with 30% that will succumb typically to metastatic disease within 5 years of that diagnosis. And there really aren't great predictors, as in there are in the dog. There are no great predictors really of who's going to be an elite responder versus a less fortunate outcome. And there's no real effective treatments for that metastatic disease when it is finally established itself in the lung or elsewhere. So as I mentioned, in the dog, we have predictable occurrence of fatal metastatic disease generally within a year. And this has been recapitulated over many, many studies over the last 25 to 30 years in the dog. And so a couple of years ago, this is, well, more than a couple, this is like 2012 or so, we partnered with the Morris Animal Foundation to launch a series of clinical trials under their Osteosarcoma Project umbrella. This was a longstanding initiative to really leverage the canine osteosarcoma patient as a model to advance anti-metastatic therapies that could benefit both dogs and people. So multi-million-dollar, multi-year initiative to really draw from the community promising agents into this clinical trial strategy. And so what I'm going to show you today is some of the data that we've generated from the contemporaneously accruing control arm. So alongside the investigational agents that we're recruiting at the same time, we had a contemporaneous control. And that's really important when we're trying to prospectively evaluate new treatments, and we want to minimize the bias of a historical control group as much as possible. So in that case, for us, we define standard of care as, again, amputation of the affected limb followed by 4 cycles of carboplatin chemotherapy. And these studies were large. They were fully randomized in a prospective design, and they had were powered to detect a 50% improvement in disease-free interval. So that's actually a pretty significant bar for us to try to reach. And many people within the osteosarcoma community were included on sort of this algorithm for agent prioritization. So how do we know which agents we should study first, second, third? So we advertised the trial, and this is the current status of the first 3 arms. So again, we have the standard of care arm, COTC-022. It was prospectively randomized against adjuvant rapamycin treatment. That study is mature, and that publication will be out probably in the next 2 to 3 months. So a total of 332 dogs were enrolled to the first 2 arms, and then the third arm will close later on this year. We've got 124 dogs in that study where they go through the standard of care, but then they receive an adjuvant form of immunotherapy in the form of a listeria, recombinant listeria HER2/neu vaccine. And that is a follow-up to this paper of Nikki Mason and Yvonne Patterson from Penn, showing in a small cohort of dogs a significant improvement in outcomes in dogs that had received that HER2-targeted listeria. And so the Morris-funded study through our program at NCI was meant to extend upon this early observation, and then with an eye of taking this agent forward into a pediatric trial. So just in the interest of time, the study schema I mentioned already, the standard of care. There's very prescriptive follow-up here. So we know exactly when dogs are assessed for metastatic disease, when they go off study. We have all of their primary tumors and their, whenever possible, metastatic lesions biobanked for initiation of further work. We had a lot of early drop off, and I think that was one of the really important points, is that you can't necessarily know a dog has osteosarcoma just by looking at its x-ray. We had some dogs that actually had an incorrect diagnosis. Some of them had metastatic disease that was defined based on their surgical exploration of regional lymph nodes. So really interesting early demographics and descriptions from those patients too. And this is not mature data, but we're getting very close to having the fully mature data set for public dissemination for those who are interested in looking at adjuvant or other new treatment options for osteosarcoma. I would definitely want you, if you're interested, please contact me, and when I can, I will disseminate this full standard of care data set to you so you can use it then to compare to your investigational agents, and we can talk about the best ways to do that. But the median DFI for this trial and this arm was only just shy of 6 months, so a little shorter than what's been previously published. Probably that's a reflection of how often these dogs were assessed clinically for disease progression. And so, I mean, this is a good example, I think, of where the human pediatric osteosarcoma and the canine osteosarcoma communities came together around a really exciting opportunity to study this disease in detail and know where the opportunities could be to benefit both types of patients. The Children's Oncology Group is a major stakeholder in all of this work, so we try whenever possible to harmonize our efforts in dogs to what is important on the COG side as well. And this HER2 new vaccine has been licensed recently to OS Therapies LLC, which is a company in Northern Virginia. So we're interested in continuing to work with them as our data matures to see whether we can take this forward in partnership with this group to see about a pediatric trial in the near future. And so finally, what I will leave you with is that when we do all these clinical trials and we enroll these hundreds of dogs across the country through a clinical trials consortium that we operate, we wind up having a really nice population of biospecimens in our biobank that are all outcome linked. And so we have recently launched the Dog Squared program, deciphering or decoding the osteosarcoma genome in dogs, with an eye towards linking all of this multi-platform omic data to what has been generated in humans under the NCI Target Initiative and others, other data sets within the community. And so we have untreated primary tumor, matched normal, a cohort of biofluids, and so we're going through the exercise of whole genome, whole exome, RNA-seq, and some methylation profiling and proteomic data to really nail down that landscape and look for outcome-linked prognostic signatures within these dogs. So we don't know really who at the beginning is, like we said in humans too, we don't really know who your elite responder and your unfortunate early treatment failure patients may be. And so the idea here is to really use the dog as a space for gene discovery and prognostication that can be applicable to both dogs and humans. And so once we generate that data, we'll bring it back to the lab and interrogate some of these new druggable targets with the available mouse, xenograft, and PDX models that we have in my lab, and then use that input from COG and others within the human osteosarcoma community to prioritize the next generation of canine clinical trials and have it be a self-perpetuating machine that really drives and is a catalyst for discovery in this space. Last, I want to leave you with a new opportunity for data sharing, which is a good topic for this meeting. The NCI is creating a canine genomics data commons, which will house canine cancer data, and it will be a publicly available open forum for you to harvest and look at analytical tools that are mostly cloud-based to look at relevance of canine cancers to their human counterparts. So if you would like more information about this, please feel free to message me. We are actively curating data sets from those in the community who have worked in canine genomics within the cancer space specifically. So if there's any interest in learning more, please let me know and I can fill you in. Okay, so we have lots to work on in the future, but I think that the main points here that we need to really continue to emphasize the scientific rigor that we place in our dog trials and our dog studies, continue to dialogue heavily with our human counterparts to make sure that everything that we do has an eye towards human translation. One thing that I didn't get a chance to talk about much, but I think is a really important point too, is developing canine-specific reagents and correlative assays to support those trial efforts is another major area of development for comparative oncology as a whole. So lots of people to thank within my program and within the NCI and the community as a whole. So thank you for all that you've done to help make sure that our work is successful and moves forward. And I'd be happy to take any questions during the panel or whenever the best time for that is. So thank you again for your attention.
All right, well, thank you so much for your time, Amy. And just to remind everyone, we are going to have a question and answer session after our next speaker. So when the panel's finished, we can ask the questions, and please use the Q&A feature that is at the bottom of your screen to send in questions.